so I know some of you are planning on taking organic chemistry, so that's what organic chemists do. They convert compounds from one functional group to another functional group, or what they do is they start with small compounds and make bigger compounds, or they take big compounds and make smaller compounds. So in many cases, it's really just converting one type of function to another type. Okay, are there any questions on quiz 2? Okay, so let's move on. I just showed this table last Wednesday. That biomolecules are organic compounds. So in this table listed 4 different types of ways biomolecules are classified: carbohydrates, proteins, lipids, and nucleic acids. The elements found in each type of biomolecule are listed here, as well as the function groups listed. We talked about some functions of these type of biomolecules, and also said that some of these biomolecules behave like polymers. For example, carbohydrates can behave like, or actually are polymers. You have the simple carbohydrates and the complex carbohydrates. So complex carbohydrates are examples of polymers. The name of the bond which holds those simple carbohydrates together in that long polymer chain is called a glycosidic bond. Proteins are long chains of amino acids, and it's the peptide bond which holds together those amino acids and proteins. Lipids, although they're very big molecules, are not considered polymers because they don't have repeating units. Okay, so note that a polymer has to have a repeating unit which is called a monomer. And finally, nucleic acids, these are also considered polymers. These nucleic acids consist of a sugar base and a phosphate, and the phosphodiester bond is the bond which holds the nucleic acids together in the long chain between DNA or RNA. Okay, so these slides are posted on the Chem1B website. Okay, so here's a couple of questions on biomolecules. Another biomolecule is important. So why is glucose important? So I give you 3 choices here. Glucose is important because it's sweet. It's metabolized by our body as a fuel. Or C is found in our blood. Okay, I should have asked this question first. How do you guys think about it?
Taking it right now.
Okay, so who says glucose is sweet A? It is sweet, but it's not as sweet as sucrose. Who says it's B? Glucose is important because it's metabolized by our body as a fuel. And who says it's C? Glucose is found in our blood. Okay, that's true too. Glucose is found in our blood. Okay, so really it's B. Glucose is important because it's metabolized by our body as a fuel. So note that even when we ingest a complex carbohydrate or a sucrose, which is disaccharide, the sucrose, which consists of glucose and fructose, that's converted to, that breaks down into glucose. So for example, there's an enzyme in the spit in our tongue which breaks the glycosidic bond, so sucrose converts to glucose, and then this starts that metabolic cycle. What's that cycle called? Krebs cycle. Yeah, Krebs cycle. Okay, so glucose then is converted to well, ATP and ADP are involved. You know, pyruvic acid. It's converted to. Oh yeah, eventually. Okay, so it goes through all those steps. It's converted to CO2 and water and energy. So in this case here, note that ATP and ADP are involved. Because some of these steps, we'll talk about this when we talk about energy and chemical reactions. See, some of these steps in this metabolic process do not occur spontaneously, so that's why ATP is involved, because ATP supplies the energy to make some of these steps of this reaction.
Oh, I'm studying this in bio right now, and there's a certain step called glycolysis.
So glycolysis is actually one of the metabolic cycles, which converts glucose to.
So something that tells us like what's NAD and NAD.
Well, NAD, that's actually a type of, you have NAD and NAD plus and NADH. So one substance is an oxidizing agent, the other one is a reducing agent. Okay, so after glycolysis, actually nothing's being oxidized, but there's another cycle after that, which involves the oxidation or reduction. I get it. Okay, let's go on to part 2. 2 forms of glucose exist in our body. So I showed the 2 forms here. So here's one form of glucose. Note that it's a chain of 6 carbons. So I have 1, 2, 3, 4, 5, 6 carbons. The other form of glucose is this ring. Okay, so we have 6 carbons in this ring. We have 1, 2, 3, 4, 5, 6. Are these 2 structures structural isomers, stereoisomers, or different compounds? Okay, so remember, what's the definition of a structural isomer? Okay, so the same chemical formula but different conductivity, like Yolanda said. What's the definition of a stereoisomer? Yeah, so same chemical formula, same conductivity, but different orientation space, like John said. And what about a different compound? Yes, a different chemical. Okay, so I'll let you think about that for a minute. So look at those structures again. We're showing the skeletal structures. Note that this one is a chain, and this one is a ring. So when you come up with an answer, share your answer with the person sitting next to you. See if you come up with the same answer. If you don't, you can arm wrestle or
The 3 of them? Oh, the 2 forms of glucose? Is it?
number 2.
Yes.
No, you guys are already talking.
Oh, okay. That's what I forget. I just don't have it.
See, that's what it's here. I know you're writing furiously, so that's what happened. So it turns out that glucose is an example of a simple carbohydrate. Okay, so simple carbohydrates are monosaccharides that have between 3 and 6 carbons. So this is actually, these glucose molecules have 6 carbons in them. Okay, so the simple carbohydrates are either the monosaccharides or the disaccharides. And the complex carbohydrates have more than 12 carbons. Okay, so how many of you think that those 2 forms of glucose are structural isomers? Sorry, so you guys are very tentative raising your hands. Let's say it again. How many think it's 1 structural isomers? Okay, here's the other. How about 2 stereoisomers? And how about 3 different compounds? So how come a lot of you aren't voting? No, because number 4, no idea isn't up there. Okay, so let's take a look at these structures. So first, is the chemical formula of these two compounds the same or different? Yeah, so if the chemical formula is the same, what is the chemical form of glucose anyway? Yeah, so it's going to be C6H12O6 for both. So these are going to be isomers. They can't be different compounds. You can eliminate that one. Are they structural or stereoisomers? So if they're structural isomers, they have the same chemical but different connectivity. Stereoisomers, they have the same connectivity but different orientation in space. So is the connectivity of these two the same? Yeah, because note here, you look right at the top, the CHO. What's the arrangement of these three atoms, CHO? In other words, what function group is CHO? Yeah, because this is shorthand for this arrangement. Yeah, so if you see this, CHO, that's the same as this. And what function group is this one? Yeah, that's the aldehyde function. Okay, so if this chain form of glucose has the aldehyde function group, you see the aldehyde function group in glucose, in this the ring form. So that means that the connectivity is different because we're seeing a functional group in this form of glucose, which we're not seeing in that one. Therefore, these two forms of glucose are what type of isomer? Structural isomer. So let's say one. Okay, question 3. Locate the glycosidic bond in sucrose and common table sugar. So sucrose consists of here's a glucose ring, and then here's a fructose molecule here. So we have the glucose and fructose. In fact, one more thing I want to mention. In our blood,